Today we're going to finish thinking about T cell development and then start talking about immune cell trafficking. So we, if you recall, have been thinking about T cell development in the thymus. And we've seen a number of different steps that are kind of T cell precursor will undergo in the process of developing into a T cell in the thymus. And we left our cells as double positive cells that are expressing both CD4 and CD8 and that are undergoing both negative and positive selection in order to make sure that we have T cells that respond to MHC, but that we do not have T cells that are going to lead to autoreactivity. And so I introduced you to the Goldilocks model of thymic selection last time, where I told you that there's sort of 4 different outcomes that can happen with a developing T cell that is a double positive here based on the signal it receives in the thymus. It can either get sort of a no-low signal. It can get a sort of intermediate on the low side signal, intermediate on the high side signal, or a very high signal. And as you can see, this sort of intermediate high, I've whited out, and we'll see more about that later, exactly what happens there. But if we get low to really no signal, that cell fails to be positively selected because it doesn't even bind to MHC. And it's going to undergo death by neglect. So it's going to undergo apoptosis, but it's not sort of actively killed. It's just sort of passively dies because it's not getting any signals for growth. The cell has a very high interaction with self-antigen. It's going to be negatively selected. And so now it is an active induction of apoptosis because we want to get rid of those potentially harmful cells. And if the cell is sort of getting, not too high, not too low, but just right, like Goldilocks signal, then it's going to be positively selected. It's getting a little bit of signal, a little bit of a pat on the head, and will survive to eventually leave the thymus as a T cell. So one of the things that is important to note here is that all of this is taking place based on interactions between those developing T cells and the other cells in the thymus that I've referred to as the structural cells or the stromal cells. So it's sort of all the other parts of the thymus. And recall that those cells, or know that those cells, are not impacted if we irradiate the mouse. So those are not impacted by radiation. And by interacting with those cells, that's how the developing T cells learn what self is. There was a question last time about thymus transplantation and the fact that if we do thymus transplantation and we use a different MHC background than the rest of the mouse, that the answer to what the T cells are restricted to is always the MHC of the thymus. Yeah, if you can do things experimentally that result in like your mouse having an entire set of T cells that are useless to it, because they don't actually bind to anything in the periphery. They think the thymus is self, and then they leave the thymus and they don't find any self there. So yes, you can do weird things like that, but that's not something that we need to get super focused on. Those structural cells of the thymus are nucleated cells. As a result, they express MHC class 2 or class 1, as do all nucleated cells. And if you remember, some of those structural cells of the thymus are on the list of specialized cells that present on MHC class 2 as well. So in the thymus, those developing T cells are able to see antigen presented on class 1 as well as antigen presented on class 2 in order to develop. And in fact, those cells are interacting with either class 1 an antigen or class 2 an antigen. There are lots of things that we do to understand T cell development. For example, remember we had our 4 different populations. First our double negatives, then our double positives, and then our 2 single positive populations. If we make a mouse that is missing MHC class 1, well, the cells come in, they're double negative, they start to become those double positives, but then they try to interact with class 1 or class 2. Class 1's not there. So none of those cells can actually become CD8 T cells. None of them that would have become a CD8 T cell that would have interacted with class 1 can get the pat on the head to be positively selected. So in this mouse, you end up with no CD8 T cells in the periphery, though you do end up with CD4 T cells in the periphery. If you made a mouse that did not have MHC class 2, yeah, you're going to have double negatives, you're going to have double positives, but then no cells can get an interaction with class 2 because it's deficient. So no cells can develop into a CD4 cell, though you will get some that are CD8s. These types of experiments, as well as many other types of experiments, are some of the ways that we actually figured out the order of these subsets, though there are many other types of experiments that allowed us to do that. If you recall when we talked about B cell development, I also told you about some experiments with B cell development where researchers genetically modified their mice to insert into their genome a pre-rearranged immunoglobulin antibody heavy chain and a pre-rearranged immunoglobulin light chain, and got mice that had 100% of their B cells and thus 100% of their antibodies being made against one antigen. Again, these mice would not do well if they ran out free in the world and got infected with pathogens, but they can be really useful for some lab purposes. We can do the same thing with T cells to make TCR transgenic mice. Here I like to sort of point out a couple of sort of key things. So if the way that we do this is we start with a T cell that we like. I really like this T cell. That T cell came from a mouse. And so when we picked that T cell that we really liked, we know what MHC molecule that T cell recognizes. In this case, this T cell recognizes, this just says H2K, but if I remember correctly, it's K of K. And we know that this T cell recognizes a particular peptide, this influenza peptide. And so when you would pick this T cell and say, I want to make a mouse where 100% of its T cells are this, or copies of this. Again, that mouse would not do well in the wild, but it's going to be a really useful lab tool. And so once I do that, I can identify the TCR alpha and TCR beta genes in this T cell that I liked so much. And I can insert pre-rearranged versions of those into the genome of a mouse. If I do that, and if I started in this case with a CD8 T cell, when those cells start to develop in the thymus, they're all going to get a good signal from MHC class 1 because they all are making that exact receptor that bounds to class 1 plus peptide. And so they're all going to be CD8 T cells. When I look at that mouse's thymus, I'll see double negatives, I'll see double positives, and I'll only see CD8 positive cells. I won't see any CD4 positive cells. And so that mouse will actually make 100% of its T cells that will be clones of that original T cell, which we can use as a nice source of these cells. If the original T cell that I picked had a receptor that recognized class 1 and was, of course, thus a CD8 T cell, and I put that into a mouse embryo and let those T cells develop, I only get CD8 T cells because those developing T cells can only interact with class 1, and we only get CD8s. If I had done the same thing, but my receptor recognized MHC class 2, because it was a CD4 T cell, I put those genes into an embryo of a mouse, and so all of the T cells that develop from that embryo will have that receptor. Those cells we're only going to get CD4 cells that are going to mature. That mouse is going to have 100% CD4 cells. So that's one thing I can do that both helps me understand some of this information about T cell development and also is an interesting lab trick. But there's one other thing that we could think about with such an experiment. Again, let's imagine I picked this T cell because I really like it so much. I originally got it from a mouse that had the H2K MHC type. And so this T cell receptor recognizes MHC of the H2K type. I'm going to insert those alpha and beta TCR genes into an embryo and then let that embryo develop. I just showed you, I would just put it into the embryo, let the cells develop. But I could take embryos that had different MHC genes. For example, an embryo that had the H2K, the same MHC as my T cell that I put in, or I could put them in embryos that had a different MHC type. If I have my receptor that responds to a particular MHC type, here it's called MHCA, and I put that into a thymus that also is MHCA, then the T cells come out. Woo! But if I were to take that same receptor that responds to a peptide being presented by MHCA and put it into the genetics of a mouse that has MHC type B, now when those T cells are developing, when they try to interact with MHC, they're not going to be able to interact with MHC. They're going to get no signal. All those developing T cells are going to try really hard, see their green cells trying really hard to get a signal. But they're not going to, because this MHC type doesn't match the MHC that they recognize. And so they're all going to die. See, look at how sad they are undergoing apoptosis here. And so in the end, this mouse isn't going to make any T cells. And so what I want you to notice from all of this is that the way that these developing T cells are really learning what self is from the stromal cells of the thymus and their interactions with the stromal cells of the thymus. And that we can actually learn quite a bit about some steps of this process. through some of these types of transgenic experiments. But there are two other really key things that we need to think about in terms of central tolerance in the thymus. One of them is where I left you last time, and it's just because it's a little bit of a thing that sort of blows everyone's mind, but it's so important to know about T-cell biology in general. And a way that we can think about this is that when we think about T cells developing, and when we think about B cells developing and their central tolerance mechanisms, in both cases, there's a T cell situation and a B cell situation. In both cases, there's something that you might notice that you're like, oh, that's really a problem. Oh, that really sets us up badly. And in both cases, you might be like, I don't know how we don't have autoimmune diseases. And the answer is really because we have peripheral tolerance too. Yay, peripheral tolerance. And hopefully both cases make you realize, oh yeah, I'm really glad there's peripheral tolerance. And so we talked a little bit about what the T cell problem was last time. And the T cell problem is that all of the T cells, in order to leave the thymus and be actual T cells in the body, have to get a little bit of signal. They have to get a little pat on the head that said, you have a good receptor, don't die. Your receptor binds MHC, don't die. And the only signals that are available in the thymus are self-antigen signals. So every T cell that comes out of the thymus has sort of a low affinity for self. It must have in order to get positively selected. So every T cell that's coming out of the thymus is a little bit self-reactive. The idea is that if it is weakly self-reactive, perhaps it will react strongly to something else, like a foreign antigen. And so every T cell is putting us right on sort of the knife's edge of autoimmunity. And if a relatively small number of things go wrong in the activation of that T cell in the periphery, we can end up with autoimmune disease. So we're very excited that we have peripheral tolerance. And so you can notice that, this scheme of how T cell development works does put us very much at risk. And every T cell in your body in the periphery is a little bit self-reactive and is being held back by peripheral tolerance mechanisms. So that's our T cell problem. We saw when we talked about central tolerance in B cells, a B cell problem. What was our B cell problem? Do you remember?
Yeah. That they weren't exposed to every cell antigen in the bone marrow?
Yeah, and so the problem with B cells is when B cells are undergoing this central tolerance process, they only get tested against antigens that are present in the bone marrow, which is like pretty good because you can imagine anything that's, everywhere in the body is going to be in the bone marrow. But they're not tested against, say, proteins that are only made in the eye. Or proteins that are only made in some specific tissue. And so our worry with B cells is, oh my gosh, what about tissue-specific antigens? The B cells are not getting tested against tissue-specific antigens. And so you might wonder what's up with tissue-specific antigens and the thymus. It turns out that the thymus actually expresses every protein in the body. So, for example, there are some proteins that are expressed only in the retina, but the thymus also makes them. There are some proteins that are only in the ovaries, but the thymus also makes them. So we don't have that same problem in T cells that we do in B cells because the thymus, those structural cells of the thymus, are really unique, and they actually transcribe every gene, translate every protein in the entire body. So every self-antigen is available in the thymus for T cells to test against. For a long time, this was sort of the answer. And right as I was starting graduate school, we actually figured out how that works. So the thymus has a transcription factor called AIRE, known as the autoimmune regulator. And AIRE is active in the thymus, and it actually allows for transcription of basically every tissue-specific protein in the body. And so this is actually some of the data from one of the studies that looked at this. And so here they were looking at proteins whose expression changed if you got rid of AIRE. And so they looked at different genes that were controlled by AIRE. And most of them you can see are red, or in this red color, which are proteins that are usually only made in one tissue of the body. And so these things that normally would be tissue specific, AIRE is able to turn on. And so you can see that AIRE allows the thymus to make spermine binding protein that's normally found in the prostate. Also allows the thymus to make insulin so that we can actually test against insulin and potentially block diabetes. You can see that we've got prepro neuropeptide Y, a brain protein. We've got oxytocin. We've got we got salivary protein 1. So we've got a lot of proteins that are made only by one part of the body. There are some other somewhat broader genes that are also seem to be regulated by AIRE, but the basic idea is that AIRE is turning on all of the transcripts in the body so that we can test our T cells against all of these tissue-specific genes. So I want you to imagine for a second what might happen in a person who doesn't have AIRE. So if you had a person who didn't have this gene AIRE, what might occur with them? Yeah, Jonah.
They probably have some autoimmune disease since the T cells couldn't recognize the specific.
So Jonah thinks that they would have some kind of autoimmune disease because we might imagine some T cell is going to get out of the thymus that shouldn't have. Some T cell should have been deleted because it had a strong signal. Now can't get a strong signal because its antigen isn't in the thymus, and so it's going to escape the thymus and potentially give that patient an autoimmune disease. Is that what you're thinking here, Jonah? Okay. Do you think that one T cell would leave and be a problem in this case? So what do you think, Brenna? No. Instead of one T cell, what's the problem?
Well, if they're all self-reactive, I think it would have to be a majority of them.
So in this case, actually, it's not just they have an autoimmune disease. It's actually they have a bunch of different T cells that all get to leave the thymus that should have originally been developed or deleted. And so they have multiple autoimmune diseases altogether because it's a failure. And this is true of a bunch of failures of central tolerance. Usually if you have a failure of central tolerance, you have many different autoimmune symptoms together because you are having so many cells that are sort of escaping this process. And that is actually how AIRE was found, was in a group of patients who had this weird phenotype of multiple autoimmune diseases. There was a genetics study done, and it was found that they were all missing this gene. And so these patients have a disease known as APECED. And so APECED patients are missing AIRE. This is how AIRE was found. I will tell you what APECED stands for. No, you do not have to write it down or no. APECED stands for autoimmune polyendocrinopathy and candidiasis ectodermal dystrophy. And that is a list of all of the different diseases that these patients tend to have. One of the things that you'll notice is on this table that you can see on the left is what percent of APECED patients had different types of autoimmune symptoms. And so what you'll notice is that almost nothing on this list is 100. So each of these patients has some autoimmune diseases, and it's a different combo of autoimmune diseases in each patient, because it's sort of a question of which T cells snuck through the cracks. And in each patient, it's probably going to be a different set of T cells. So you can see that many of them particularly have issues with endocrine glands, like immune attack of the thyroid, adrenal glands. Some of them will have diabetes, will have testicular atrophy or ovarian failure. Weirdly enough, most of them get a lot of yeast infections, so the candidiasis is 100%. Some of them will get other skin or hair issues like alopecia or vitiligo. Some of them have some issues with their dental enamel, and a large number of them have a weird fingernail phenotype of nail dystrophy that you can see here. And so looking at these patients who have this weird sort of constellation of autoimmune diseases is how we found this gene AIRE. And so T cells don't have the same issue as B cells in that we do test our T cells against every single self-antigen. But they do have the problem that they all had to get some signal, and so they're all a little bit self-reactive. And so you can see that these steps of negative and positive selection are happening both in the cortex, but especially in the medulla. AIRE is present in cells of the medulla to make all of those transcripts. And so we're getting a lot of negative selection in the medulla. This is why the medulla is so much less densely full of cells. And you can see this same thing here. Here we are at our double positive. The cells may die. They're moving towards the medulla. Not so many of them are going to make it to the medulla because so many of them have died. And these are sort of our mature double positive T cells. But now our T cell needs to make one final call, which is whether it's going to become a CD4 or a CD8 T cell. So we before had CD4, CD8 double positives, and now the cell needs to actually become a CD4 positive only or a CD8 positive only, and be ready to leave the thymus. And so you can see if that cell undergoes positive selection, it gets its pat on the head. It makes the choice of either becoming CD4 or CD8 single positive. And now it's going to actually have some kind of function, leave the thymus. There have been a lot of debates about how developing T cells make this choice of CD4 positive or CD8 positive. One idea was that somehow they could tell if they were getting better signals from class 1 or class 2, and so that would tell them, should you keep CD4 or CD8? There was some idea that it was really that the cell just guessed. We now have this there's a newer model where we actually have found out that the cell actually basically is finding out whether it gets a better signal from class 1 or class 2. There's something known as a kinetic signaling model. It's basically about how long it gets signal that tells it whether it's getting signal from class 1 or class 2. If you'd like to know more about the model, we interviewed the person who did the work to understand this model on our podcast, and so this is the episode. the experiment he did was like so cool, and I'm just like, I have no idea how you ever thought of that experiment. Like, no idea. But it does seem as though the cells do find out, does my T cell receptor that I've spent so much time making work better with CD4 or CD8? And actually get those cells will then get instructed, should I keep CD4 on my surface or should I keep CD8 on my surface and stop making the other one? Yeah, Jonah.
In that case, would it be that it got a stronger signal for one of them? Or
We'll say yes, it got a strong. So basically what seems to happen is that there are time periods where the cell tries each one individually, and it's a question of, did you get good signal during that time period? Is what they showed here. And so what they actually did is they took the CD4 and the CD8 genes and they swapped which one was given by which promoter. So it swapped with the timing of it. And you could see that basically if you got good signal early, you made the thing that was under the CD8 promoter. And if you got good signal late, you made the thing that was under the CD4 promoter, even if they switched what was under which promoter. It was a very cool flip-flop experiment, is what they called it. So that's why we referred to it like that. Once the T cell has made a decision about whether to be a CD4 positive cell or a CD8 positive cell, that cell will now exit the thymus and will move to secondary lymphoid organs and be in the periphery. We can look at plots like this to know some things about those T cells in the periphery. Typically, what you might notice if you look, say, at old exam questions or things like that, is sometimes I'll ask you about looking at a plot of something that might have happened in the thymus. And sometimes I will look at a plot of something that happens in the periphery. Usually we're going to look at either the lymph nodes or the spleen. And so the lymph nodes or the spleen are two of our key secondary lymphoid organs that often represent the periphery. And so this is actually published data of T cells in the periphery, either in the lymph nodes or in the spleen. Again, you can see that we're looking at CD8 versus CD4 plots, just like we were looking at in the thymus. What you should notice here is that if we look at the percentages of cells, the percentage of cells that is double positive, has both CD4 and CD8 together, is very low. In fact, if I was going to draw this plot, I would drop chalk on the ground. One of the biggest differences I would have between a thymus plot and a plot of something in the periphery like a lymph node was that in the thymus I'd have a lot of double positives, and in the periphery I would have none. I just wouldn't even draw a population there. It actually surprises me that they found that many in this paper. So if you remember, in the thymus we had a small population of double negatives, sort of medium populations of CD4s and CD8s, a big population of double positives. In the lymph node, we're going to have CD8 single positives. We're going to have CD4 single positives. They've all made up their mind. They're not expressing both CD4 and CD8 anymore. They all chose. And then we can think about what's going on in this quadrant here, which is the CD8 negative CD4 quadrant. If I do an experiment like the experiments that were done in these figures, you can see that we do see a nice population of double negative cells. Those cells might be some of our NK cells, they might be some of our B cells, they might be some of our gamma delta T cells, they might be some of all of our non-T cells, which are also found in the secondary lymphoid organ. If I do the same plot, but I gate on CD3, and gating, remember CD3 was part of the co-receptor that's important for signaling in the T cell receptor. CD3 is really only on our T cells, and we'll say for our purposes on our alpha beta T cells. If gating on CD3 means that I'm only examining cells that have CD3. I did some earlier steps of my flow cytometry to eliminate cells that did not have CD3. In that case, I'm only going to see the single positive populations, the CD4s and the CD8s. I'm not going to see the double negative population because I have eliminated them by eliminating my cells who do not have CD3. I don't have to worry about gating on CD3 in the thymus because we really don't tend to see a lot of non-T cells in the thymus. And so I see the same thing whether I gate on CD3 or not in the thymus, but in the periphery, those other cells, like, again, B cells, would make a big difference in what I might see here. So this is a very exciting part of the semester because we have now developed all of our cells. And we are now finally ready to be in the periphery making an adaptive immune response. We are going to now do something about a microbe instead of just preparing for that microbe. And so remember, we had our B cells undergoing B cell development in the bone marrow. Well, now they're out in the periphery. We had our T cells undergoing T cell development in the thymus. Now we're out in the periphery, largely thinking about things that are in the blood, things that are in the lymph node or other secondary lymphoid organs like the spleen, and potentially things that are in other organs like, say, the respiratory tract or the GI tract or the skin or something like that. So now we're out in all of these other organs, sort of in the rest of the body. And we can kind of come up with some ideas of where exactly these cells are. Different versions of this image from different places sometimes give us wacky numbers. So I'm not going to super focus on them. But what I want you to notice is that this is telling us that I believe this wants us to say that 50% are in the spleen, 40% are in the lymph node, and 10% are elsewhere. Here you can also see, some of them are leaving the blood and going into mucosal tissue, and they're saying that's 10%. Like 45% they're saying is in the spleen, 42% is in the lymph nodes. Some of those cells are going to travel between organs in the blood, so some fraction of them are in the blood at any given time. Some of them are going to travel through the lymphatic vessels, so some of them are in the lymph fluid at any given time. You can see some of them are just in other tissues. It's really hard to say how many of them are in other tissues. because it would require you to like grind up those other tissues, isolate every cell, and count. And I personally am not volunteering to be the person whose skin they grind up or whatever. So the numbers that people estimate have crazy variation. But you can also see some interesting information about timing here. If the cell heads to the spleen, it's usually in the spleen for like maybe 5 hours. And that's honestly if it's not tending to make a response. It's going to go to the spleen and like look for antigen. If it starts to actually find antigen and do something in the spleen, it's going to be there a whole lot longer than 5 hours. It's going to be there for days. But it might go to the spleen, check the spleen for antigen for 5 hours. It might go to the lymph nodes and check lymph nodes for antigen, and it might be there for like 12 hours. If it makes a full circuit in the blood, so it goes from heart all the way around the blood vessels of the whole body and back to the heart, that takes it 30 minutes to just circle around the body in the blood. So when we talk about the periphery, there are a lot of places that we might be thinking about with our lymphocytes. But we can also think about sort of a little bit about what our response is going to look like with our lymphocytes. In the figure in the upper left, this is what I often think about as a really classic figure for this. It's from your textbook. Every time I ever teach this, people get mad at that figure because they're like, I need more specific numbers. I don't like it that it's that vague. And they're like, I want more specific numbers. So here is a piece of data that I published that has more specific numbers. But I will tell you that the reason why we usually don't show it with those numbers is that things like the dose of the pathogen, the specific which pathogen you use, how much pathogen dose you use, all that kind of stuff changes the specific numbers. But if you want with numbers, there you go. And the key thing that I want you to notice about either of these images, in both cases on the y-axis, we are looking at an adaptive response in the periphery, is where the infection is relative to where we actually start to measure the adaptive immune response. And so here you can see in our sort of generalized version that we're starting to see those lymphocytes in the periphery make a response at about a week following infection. And you can see when I actually did this experiment, at day 7, I could sort of start to see stuff. Things weren't super interesting until like day 10. So it's usually about a week. Those lymphocytes were in the periphery that whole time. Wasn't like you were waiting for them to finish development. They've been in the periphery. And so the real question that we want to think about for our adaptive immune response is what the heck is happening during this week? What's taking so long? I would like my lymphocytes to stop being so lazy and protect me earlier. What kinds of things have to happen during this period of time? And so for some of the next lectures, we're going to be talking about some of the events that are happening in this period of time. You can see 2 different versions of this at the bottom. And so one of the things that has to happen is that we need to actually get the antigen to a lymphoid organ. The first place that you get an infection isn't usually your spleen or your lymph node. And so we have to first get the antigen to the spleen and the lymph node. So you can see secondary lymphoid tissue, close to infected tissue is made aware. Then we've also got to get the B and T cells there. We have to get the lymphocytes to that same lymph node. So you'll notice that there's sort of this transportation problem. We then actually have to have signal transduction happening And the B cells and T cells actually do something with some recognition. They need to actually do some processes of expansion and differentiation. So they're going to change and gain new properties. And then you can see, oh my gosh, this one also says there's more travel. They now have to leave the lymph node and say, go to the lung in your SARS-CoV-2 infection and now actually kill the SARS-CoV-2. So we had to both bring the antigen to the lymph node and be like, hey, T cells, please learn about this thing. We have to bring the T cells there so they can do the learning. And then we have to send the T cells back out to actually go kill the microbe. And so what I want you to notice here is that travel is a part of this, though there are other parts of this. And so for the rest of today, I'm going to be talking about some of those travel details. And that's where I will also be talking about things on Wednesday, although I will also then go into some of the activation. And so again, if we're just thinking about what the heck takes so long, one of the parts of this is this phenomenon called trafficking. And so you can see that trafficking might take up to 24 hours. Here you can see that happening with, in T cells, we're often going to see at least 24 hours for that trafficking to happen. So between when the microbe first comes into your body and when it even gets to the location for this response to happen, you've got about 24 hours. It is a bit faster with B cells. And then you can see, again, all of these additional events taking some amount of time. If you look back at this little time scale from your textbook, the other thing I want you to notice is that we kind of have to deal with how the antigen gets transported and how the T cells get transported, because they both have to meet up in that lymph node or secondary lymphoid organ in order to start the response and to do the next steps here. So I want to tell you a little bit about how antigen travels right now, and then we'll think about how the T cells travel after that. There are a couple of ways that antigens can go to the lymph node. One of the ways that antigens can go to the lymph node is just that we have these nice open lymphatic vessels throughout the body that might pick up free antigen. So here you can see on the right from your textbook, here's this nice open lymphatic vessel, and random microbes or random antigens might just get pushed in there when they're free in the interstitial fluid and sort of passively get pushed through the lymph. And you can see that happening, some of these little red dots going through this lymph freely. You can also see all these little red dots going freely. That's one way, it's sort of a just transport in the lymph. We can also have situations where we might have cells that are in that local tissue that might grab antigen and travel and carry that antigen to the lymph node. And so we can also have local cells in the tissue that pick up antigen and carry that antigen to the lymph node. And you can see that in both of these cases as well. You can see that we might carry the pathogen or carry the antigen to the lymph node, or it just might go in free through the lymph. Generally, the type of cell that is hanging out in the tissue that is going to that's going to carry antigen to the lymph node is a dendritic cell. Dendritic cells are very closely related to monocytes and macrophages. They are cells of the innate immune system. They really do all the same kinds of things that you can think about as macrophages that we talked about before. But a big part of what we think about with dendritic cells is their ability to pick up antigens and move them to the lymph node. And so dendritic cells are sometimes thought about as being like the bridge between innate and adaptive immunity. They're really key, even though they are 100% innate immune cells. They are really key in helping us turn on adaptive immune responses. In particular, we really need them for turning on T cells. When we're in some kind of tissue, like this foot that stepped on a nail, that dendritic cell is an immature dendritic cell. And you can see that immature dendritic cell here. For all intents and purposes, that immature dendritic cell looks like what you saw in lab 1 as a monocyte. And frankly, for our purposes, it's a monocyte. But that dendritic cell can become activated, can mature, and can just start look more like what you see here. And what I want you to notice is all of these little tiny pieces coming off of the membrane, like little tiny little fingers. This is why these cells were named dendritic cells. People originally were like, oh, they look like they have little things coming off of them, like cells in the brain. So we don't know that they're different. Well, we'll just give them this name. There are a couple cells that we're going to learn about where you're like, seriously, people, those names don't fit at all. These are not brain cells. They are in no way related to the brain. I have actually been in situations where I tried to say something about a dendritic cell to a physician, and he thought I was saying that I was talking about the brain, and he somehow took. I was like, well, this is what happens in this vaccine with the dendritic cell, and he's like, you mean it causes multiple sclerosis and kills the brain? And I was like, no, dendritic cell, not brain, different things. So you can see that our dendritic cell is going to change its morphology and really get these projections. And the big thing about that is that that's giving the cell a lot more surface area, both surface area that it could use to potentially pick up antigen, but more importantly for us, surface area it could use to turn on a T cell. Surface area, extra surface area where it can present stuff to T cells because now it has more membrane space. What you can also notice is that if this fancy looking mature dendritic cell tried to travel through the blood, which I'll be saying more about in a little bit, it would get torn up. The cells that are in the blood tend to be nice, perfectly circular cells that are not likely to get pieces torn off of them, while this dendritic cell would totally get pieces torn off of it. And so as our, sorry, Brenna, do you have a question?
Oh, I was just confused on the location.
Okay.
So, like, if we're talking about the foot and the nail,
Mm-hmm.
the immature would be found in the tissue.
Yep.
Then would immature also be found in the blood?
So let me show you what's going to happen.
Oh.
Okay, perfect. So our dendritic cell is going to become activated. And it's really going to become activated by seeing some kind of PAMP. So it's going to switch from immature to mature. It's probably going to make that switch, Brenna, in the foot when the microbe comes in. So it was hanging out, resting, immature, in the foot. Then a microbe came in, had some MAMPs that could stimulate PRRs, and that's going to activate the dendritic cell. And so you can see, again, in this on the view on the left, here's my dendritic cell that was immature. Now it's going to get some stimulation, and it's going to be a mature dendritic cell. It's going to have way more surface area, these big, long projections, and that's going to let it put way more proteins on its surface so that it can talk to T cells. When that dendritic cell will at the beginning it will be doing lots of phagocytosis, trying to take in whatever it finds. When it gets activated, it's not going to be doing phagocytosis anymore. It's like, I already caught something. I don't need to catch anything else. It's going to suddenly start expressing way more MHC, especially MHC class 2. And it's also going to move and start moving towards the lymph node. And it's going to move there, Brenna, through the lymphatics, not through the blood. And so you can see this happening here as well. So our immature dendritic cell is sort of hanging out in tissue. It's doing some phagocytosis. If it finds something, it's going to start maturing and head through the lymphatic vessel to get to the lymph node so it can start talking to T cells. And it's going to have changed its phenotype. It's going to have more proteins on its surface and more surface. And it's going to not really be doing much phagocytosis anymore. And so you can see that dendritic cell making that movement. You can see that our dendritic cell is going to start having all sorts of stuff on its surface in order to activate T cells. This is really critical because all of these proteins all over the surface of dendritic cells allow them to be the best at activating T cells. So of all the possible cells that can turn on a T cell, dendritic cells are by far the best. And it's just because they have so much surface area and so many proteins on their surface area that ideally we want stuff to be presented on a dendritic cell. One thing that I know sometimes somebody asked me previously, you can imagine, say, a virus that might infect this dendritic cell, and that being how we present on class 1, those viral antigens. Dendritic cells also might be picking up fragments of dead cells. And so if a virus killed a cell, it's going to be like there's going to be trash from the infected cell around. The dendritic cell can phagocytose that, and that's going to allow presentation on class 2. And so you will see both class 1 and class 2 presentation from some of those viral antigens.
Why are they called Langerhans cells?
So Langerhans cells are the name for dendritic cells that live in the skin. And they were named that by microscopists before we knew what a dendritic cell was. So they were just giving names to all the cells in the skin. But now we know that Langerhans cells are dendritic cells in the skin. And so that dendritic cell will go to whatever lymph node is the draining lymph node for that location in the body. So basically the dendritic cell is going to be hanging out somewhere in the body. It's going to pick up antigen and it's going to go to whatever the nearest lymph node is. And the idea, again, remember with the lymphatic system is that we have these lymph nodes in all different areas of the body. Each one kind of is responsible for a particular region and for allowing us to do surveillance of that region. So the dendritic cell is going to go to whatever lymph node is responsible for the region where it was hanging out. It's really going to be coming in through a lymphatic. It's not coming in through a blood vessel. And we sort of mentioned that because it has those big lung processes, it would not do well in a blood vessel, but it can be okay in a lymphatic. So it's going to come in through a lymphatic, which also would be how the free antigen is coming in. And what you'll notice is that the lymph node has a bunch of different types of areas. And so those different antigens and those dendritic cells can actually go to a few different areas in the lymph node. So that's how we get the antigen in the lymph node. But we also need to get the T cell in the lymph node. Because the goal here is for the antigen and the T cell to meet and make a response. Naive T cells, and so we're going to be imagining right now a T cell that has come out of the thymus and is just going around the body trying to find its antigen. So it has never actually seen its antigen before in the periphery. It's a naive T cell, have not made a response. That T cell can get to the lymph node through the lymphatics. So if it starts out, if it decides to hang out in a lymph node, it can just go from lymph node to lymph node through the lymphatics, or it can come in through the blood. Though what I'm going to show you for this process is going to be I'm going to really show you the blood process, but they can actually be coming through the lymphatics as well. This process that I'm going to tell you about of these T cells coming to the lymph nodes using the blood is a sort of specific example of a broad process called leukocyte trafficking. So we're thinking right now about those T cells coming in from arteries and veins and going into the lymph node. But the same process that we are actually going to see, which involves a T cell actually being in the blood and then leaving a blood vessel, is actually the same process that neutrophils use if they want to leave a blood vessel and go to a site of inflammation. It's also the same process that a T cell might do if it decides when it goes and kills the virus in the lung. So this overall process of leukocyte trafficking, or how a leukocyte leaves the blood vessel and goes into some other place, is a very general process. The same steps happen, no matter which example we are using. One other thing I just want to point out, it's going to be a process. It's going to have some steps. We'll see those steps next time. But I want to tell you one thing about this process, and this will sort of help us transition nicely to Wednesday. If you think about how fast something moves, it turns out that actually thinking about how fast things move is also kind of important to think about how big the thing is. Like, if I moved at like 100 miles an hour, that would be far more impressive than if the Earth moved at 100 miles an hour, because the Earth's really big and I'm really small, right? It's sort of like, how many body lengths do you move in an hour? We actually look at how quickly blood cells are moving through the blood compared to their size. They are moving exceptionally, exceptionally fast. There are some techniques that allow us to actually watch blood cells in a living organism move. I'm not going to go into how we do that, but we can actually watch cells with a microscope in the blood vessel of a mouse, which is what is happening here. And so I want you to look at how fast those blood cells are moving. Particularly think about how fast that is relative to their size. You can see sometimes if they're out in the tissue, they're going to move up maybe a little slower, but this is really what we're seeing with blood cells moving through the blood vessel. They show all sorts of different things going on in this video. So you can actually see, for example, the mouse shutter moves. There also is a place where they actually clip off the vessel, so you can watch the cells stop, and then they let them go again, which I don't know if we're going to see that long. But what I want you to think about is you can't just be in that and be like, oh wait, I'm going to stop and leave right here. Again, I love to think about this in terms of being on 95 going really fast, driving really fast. You can't be in the middle of 95 in a middle lane and be like, oh wait, I'm going to stop here, and just stop, right? And be like, I'm leaving now. You got to go through a whole process. Because if you didn't, now I think we've stopped. I don't know, whatever. If you just stopped in the middle of that flow, what would happen to you? You get trampled. You have all of the others moving through that process smash into you. and you would actually get torn in half. This is why we only have circular cells in the blood, because if they're not circular, they're not going to make it through this. But this is why we're going to need the process that we see on Wednesday, because we can't just easily leave this stream. But blood cells have the ability to go to a lymph node, go to a site of inflammation or something like that, by getting out of a stream that looks like this. So I will see you guys on Zoom on Wednesday when we will talk more about this process.